look at it, review it later, and students that weren't able to come will also be able to view it. And so this is
Okay, perfect.
our final exam review. I can't believe it's that time already, but we are here and I wanted to do this just to go over a few things and then to go over any questions that you might have pertaining to the final exam. And so I'm going to share my screen. Okay. All right. So are you able to see the screen, Cheryl?
Yes, I can see it.
Awesome. Okay. So we are in week 7 right now, but next week is week 8, which is our final week. And so I wanted to go over a few things to give you a heads up, few of the due dates and everything. So we have, as always, our discussion post. We have our lab quiz. Number 4 is our last lab quiz. It's going to be due next Friday, and then our final exam also due next Friday. Both of those are at 11:59, but the lab is at 11:55. It's also due on next Friday. And so for the final exam covers material, excuse me, from week 5, week 6, weeks, and week 7. There's no new material for week 8. Week 8, we're just finishing up labs and final quizzes and so forth. And so there's no new material for week 8 that will be on the final, but it does cover everything from 5 to 7, okay, including this week. And so there are 150 questions for the final exam. And so I strongly suggest that you do not wait till the last few minutes to try to do the exam. I want to make sure that you take your time and that you think through the problems carefully. But the way that I would suggest that you study for this exam, because it does cover a lot of material for those 3 weeks. So if you go to each week and go to the resources section, the same thing that I suggested for the midterm, but there are so many good videos here that will help you understand the material and also give you practice problems to prepare you for this final. And so we're going to go through a couple of those, okay, from each week. All right. So in this first one, this is the presentation of oxidation for oxidizing alcohols. This is actually a video. All right. And so this, once you click the arrows, then the video does begin. Okay. When you're going through these different practice problems, okay, sorry. When you're going through all these different practice problems, what I would suggest is to take notes, okay, and then review those notes to help you study. And I would also suggest that you not wait till next week to start studying. So start going through your different resources from week 5. That one's the video. These I do believe are just the notes. So reactions of alcohols with aldehydes and ketones. And so when you're going through these different reactions, they give you specific examples, but there are patterns that each of these different reactions follow. Like, for example, you have here an aldehyde, and you're reacting with an alcohol in the presence of H plus, and then you're going to get this group, okay, with your COCH3OH. When you have these different types of reactions, this gives you a specific example of the actual example of a compound, but it follows the same pattern no matter what you're starting with. And so anytime you have an aldehyde or a ketone, you react it with an alcohol, you have this basic portion, okay? That's the portion of the aldehyde and ketone. The O is going to attach to a hydrogen, and the carbon that's in that middle that was attached to the double bond O is going to have this portion. All right, so the hydrogen from here will attach to the oxygen, and then the rest will attach to the carbon. And so it doesn't matter how many carbon chains are on the alcohol. It doesn't matter if it's an aldehyde or ketone. It's still going to follow this basic structure. So when you're studying these organic reactions, you're going to look for the basic pattern. And you see how there are different colors here. That's to help you show you where everything was originally on the reactant side and where it goes to on the product side. And so you don't want to memorize your reactions based on the substances, but you want to see the pattern that takes place. And that's true for all organic reaction. You're looking for a specific pattern. And I like how in these notes, it shows you the different colors so you can look at how the pattern is. So you can look at what the pattern is for these different reactions. And so you can see that if we look at different examples, you see how this is an aldehyde, but then we have an example of a ketone, and then we have another example of a ketone. I'm sorry, we have an aldehyde, another aldehyde, and a ketone. But if we look at this, the pattern is still the same. So you still have what you start with, okay, whether it be an aldehyde or a ketone, and the alcohol is still going to separate in the same way. The hydrogen is going to go to the oxygen, and then this OCH3 is going to go on the other side of the carbon. And so when you start to look at how the patterns are, it will help you when you get to the test and you look at the reaction and you say, wait a minute, that's an aldehyde with an alcohol. I know how that's going to be structured. Or that's a ketone with an alcohol. I know how the structure is going to be. So you're able to pick the correct product from that. And the test is multiple choice. And so you can take what you've learned with the patterns and apply it to whatever reaction you're looking at. Okay. All right. And this is just an example of a cyclic. So if we look at the cyclic and again, we're looking at the pattern. And so we have this OH here. This is where you have an aldehyde and an alcohol, but it's all together in one particular compound. And so this forms a cyclic. And so what happens is the hydrogen from the alcohol still goes to the oxygen that was in the double bond. So see, the pattern still remains the same, except for this oxygen still is going to connect to the carbon, but it's not going to break away from that chain, and it's going to form a cyclic group. Okay. And so we have another example. Here we have our group, our hemiacetal. Okay, so now we're starting with that. We're pairing it up with an alcohol. And now the OCH2CH3 now attaches to the carbon that had the OH, and the OH now attaches to the hydrogen from the alcohol. Okay, so now we're taking what was the product from the previous examples, we're reacting it with an alcohol, and now we get another O group, what we call the OC group, or you may see it as an OR group, and that attaches to there. And so as you're going through these notes, I want you to notice the patterns that are formed in these different reactions. And they make it nice in these notes because they keep the colors different. so you can see where the compounds go, okay? And then there's always practice questions. So you go through and strongly suggest that you try these practice questions. Try it without clicking to reveal the answer first. And then if you didn't get it right, go back through those slides, try it again, and see if you understand why it has the answer it has. And so if we look at this, we have this particular compound, which we have a C double bond O and two C's attached to it. So that tells us it's a ketone. We know with the ketone in an alcohol, we have the hydrogen is going to go to this oxygen, and the OCH3 is going to go here. All right. And so we have two of them though. So that means it's first going to form a hemiacetal. Okay, it's going to form that first. But with the second alcohol, it's going to form the acetal. Okay, so your answer would be B. If there was just one alcohol here, then it would be the hemiacetal. Right? But because there are two alcohols, it's going to be the acetal. So it's going to look similar to this example, where first your hemiacetal was formed, but then you add another alcohol group to it, and then you form the acetal, okay? And so that's what we see here in this example. Right. So that's just to show you the patterns that you're looking for, how we go about doing or studying organic reactions. Are there any questions on that example?
Not right now.
Okay. All right. And so then there's others that are in this particular chapter. There is the oxidation of aldehydes, preparation of alcohols, ethers, and sulfur compounds, the reaction of alcohols and thiols, monosaccharides in their cyclic form. Anytime you see the word cyclic, that means it's going to be a shape, okay, that's associated with it. And then the reaction of monosaccharides. So that would be for week 5. Okay. So we're going to go back. All right. And we're going to go to week 6. All right. So in week 6, the same thing. We want to go to our week 6 resources. Okay. Before we do that, I want to make sure Yvette has joined us. Yvette, are you there? Let me stop sharing for a minute. Yvette, can you hear me? Okay. I see she's there, so hopefully she is hearing it. So we're going to go back to the screen. Okay. All right. So with week 6, just like we saw with week 5, there are some that have videos. So there's one on peptide bonds, one on amino acids. So those are just to give you a little bit more instruction as to what each of those sections are. And so go through those videos because they are helpful in understanding those different functions, okay? The peptide bonds, the amino acids. And so I want to go through, I think this is the first one that's not a video. Yeah, okay. So this is triglycerides. And with triglycerides, we're looking at those fatty acids and glycerols that combine to form your triglycerides. So if we look at the acetic acid, and there are three of them. Okay, these are our three fatty acids. This is the structure for glycerol. And when we combine them together, we get a triglyceride. And so if we look at this is a bit different. This doesn't show the, let's see if it does. Oh, it does a little later. Okay. In this example, it does show the colors and where everything is going, but let me show you the pattern here. And so you have your CH3, C double bond O, OH, and you have your OCH3. So the hydrogen from your glycerol is going to combine with the OH group from your acetic acid. Make sure that's showing that. Yeah. Does it show it on there? Yeah. All right. So the hydrogen will pair it with the OH, and that'll give you your three waters. And then the OCH3 is going to connect to the carbon in the C double bond O, and that's what forms your triglyceride, okay? And it shows it better here, but it's kind of small. I don't know if you guys can see the difference in these colors, but the OH here is in black and the H here is in black. That's to show you the things that come together to form the water. That OCH3 is what's going to connect to your C double bond O to form the triglycerides. So again, as you're looking at these different reactions, you want to follow what the pattern is, or you want to know what the pattern is. Different reactions that can be formed with triglycerides, the catalytic hydrogenation. Okay, this is a reduction reaction. And then oxidation. So with the catalytic hydrogenation, you have your alkene with hydrogen. Hydrogen for this particular reaction is always paired up with PT, which is the symbol for platinum. Okay. The reaction actually takes place on a piece of platinum metal, right? And you're going to form your alkane. With oxidation, you have your alkene plus oxygen, and it's going to form an aldehyde or carboxylic acid. And then saponification, that's the making of soaps. You have your ester hydrolysis in the presence of OH minus. So for this particular test, you are going to look at, for this final exam, you are going to look at different reactions. And so you are going to need to know if we have an alkene plus hydrogen in the presence of platinum, then you're going to form an alkane. You may not see it like this. You may see an example where they actually give you the formulas for an alkene. They give you the H2 and platinum, and then you'll look for the formula that matches an alkane. And so know these functional groups, because it may not be spelled out as alkene going to alkane, but it may give you an actual structure that's an example of an alkene. So make sure you know what an alkane looks like, what an alkene looks like, what an alkyne looks like, your aldehydes, your carboxylic acids, your esters, your ethers. You want to know what all of those different functional groups look like. And if you need to refresh, I do believe it was week 4 where it went into the different functional groups, or you can always Google the organic functional groups and it will give you, sometimes you'll see charts, okay, and they'll give you what the basic structure looks like for the different functional groups. That is always helpful to study, especially for this final exam, because a lot of the questions expect you to realize that, um, what those different functional groups are. So make sure that you do refresh that information. All right. And so if we look at what this looks like, so this is an example of an alkene. Remember, alkenes have that double bond between the two carbons. And so your alkene plus H2, platinum on the previous example, you see how it was H2/PT. More than likely, you'll see PT above the arrow, okay? So that just tells you you're in the presence of platinum. And so you see how the two hydrogens go to the carbons and break that double bond, and that causes it to be an alkane, right? So this is another example. So this would be your triglyceride. And you see with each of your triglycerides, there is that C double bond C. So we're still that alkene group. We have 4 hydrogens, and there are 4 H2s because there are 4 C double bond C groups that you want to turn to alkanes. And so still in the presence of platinum, so you may see the H2 above the arrow, and you know that everywhere you see that C double bond C, the hydrogen is going to add to it. And so you have your alkane chains where it used to be alkene chains, okay? So it doesn't matter the structure. It could be something simple like this or something much more complex. But when you see these C double bond Cs in the presence of H2 and platinum, you know that they're going to turn into alkanes, okay? So again, you're always looking for the pattern. Here again, you're looking for that pattern. And so this time, only 2 of them turned into alkanes. Okay, this was a partial hydrogenation. And this process is used for making vegetable oil, just to give you a heads up for that, right? And so you still have some double bonds, but some of them were converted into alkanes. All right. And then some examples where you have this one and this one. These are the ones that are changing, but you also have a rotation. And this is where you have your cis and trans fatty acids, okay? This is your unsaturated vegetable oils that are converted to cis and trans fatty acids. So I don't know if you've heard of trans fats, but this is the process of making a trans fat. And so these are just different examples that you will see. Okay? Auto-oxidation, this is where your alkene reacts with oxygen in the air, and you're going to get your alkane. Okay, and so this is, sorry, let me get alkane. Let me give you a, nope, that's the, all right, so you have your double bonds. I'm trying to follow the patterns here. So this separates, and you have this first structure where you have your C double bond O with the H. And that is from the top one. There we go. Okay. So this is the top one, is where it comes to here. The second one is what forms here. And the third one is there. Okay. So that's a little bit more complex. But you just, sometimes you have to take a minute and see the correlations with those. And then the saponification, and that is to make soap. Okay, that's the production of soap. And so you have your simple one that's an example, so your methyl acetate. And so you have this structure here with your NaOH. The OH, okay, if you can follow this, your O with the Na is going to go here, connect to the carbon, and then the H is going to go with the OCH3, okay, and give you your alcohol. So you have your acetate and your alcohol. And so if we look at this example, so you still have your triglyceride here with NaOH. And so this OCH2, OCH, and OCH2 group is going to go here, and it's going to combine with the hydrogens. So you have your hydrogens here. Your ONA is going to connect where that structure used to be, okay? And so it's going to separate into three separate structures. All right. Oh. So there we go. So which of the following reactions is responsible for the smell of rancid butter? Okay. So we know it's not going to be the soap, but it's the auto oxidation. Okay. And it talked about that back on one of these slides. There we go. Triglycerides have spoiled or gone rancid. That's your auto oxidation. Okay. And then the next one, which of the following reactions can raise boiling point of triglycerides, making it more solid? Okay. And so that would be your saponification. And I do believe it talked about that back here. No, I thought it did talk about it. Maybe I'm missing it. No, it didn't. All right. Oh, thought I did see that. What is it? It shows it in the glycerol. Okay, but know that. All right. So there are any questions so far?
Not right now.
Not right now? All right, awesome. All right, so let me go back and we are going to go now to this down. Oops, not that. That's not what I want to do. Okay, thank you. Go back here. Here. Okay. So we're going to go back and now we're going to look at week 7, which is our current week here. And this week we're working with nucleic acids and metabolism. And so for this one, we have a couple of videos that you can look at. I do believe that one's a video, if I'm not mistaken. Yeah, all right. So that gives you an overview of fats. And then your DNA replication and your transcription RNA. This is what we're working on in lab. And so keep in mind that you do have a lab video for that. I did give you a lab video on how to set up the lab and work through that. But we're going to look at nucleic acids. Okay. So the building blocks to all living organisms, your nucleotides, they're made from phosphates, monosaccharides, and organic base. And so this gives you an overview of your phosphates, okay, and what they look like. So they come from phosphoric acid. So you can have dihydrogen phosphate, or just the phosphate ion. So each of those are removing one hydrogen. And so if we look at that in the presence with an alcohol, so remember, anytime you have OH with just a carbon group, that's your alcohol. And so here you see, excuse me, the pattern of where things are going. So the OH from the phosphate, the hydrogen from the alcohol are going to form water. And then you get your phosphate monoester, okay? If you have two OH groups, okay, in the presence of two alcohols, you'll get your phosphate diester. So monoester, meaning you have one OCH3 group, and diester, two OCH3 groups. And keep in mind, for these examples, we've had an alcohol with one carbon chain. I think I did an example with two carbon chains, but it could be, it doesn't have to be just one carbon or two carbon. It could be more than that. The ester portion, though, is the fact that it's oxygen connected to a carbon group that is attached to the phosphorus ion. And so the phosphate ion. And so that's what makes it that phosphate ester. If we look at monosaccharides, so we have D ribose and D2 deoxyribose. They're incorporated into your nucleotides and they appear as the beta furanose form. Okay, furan, okay, is that carbon, two carbon groups. Okay, so at these points, these are carbon groups and they are directly attached to an oxygen. So there's no other carbon here. It's the carbon that forms a chain with the oxygen within that, okay? And that gives us that furan ring, right? And so the beta, whether it's beta D or beta D2, it just has to do with the location of where your OH groups are located at. And so then we see another reaction with this. With this same pattern, it's a bigger alcohol group, okay, more complex, but the same pattern applies. So your OH from your phosphate is going to leave your hydrogen, okay, from your alcohol is going to leave and it's going to form water. Your phosphate group is going to attach at the oxygen level. And so now this whole portion becomes your monoester, okay? And so it could be simple like we saw in this example, or it could be more complex like we see in this example, but the pattern remains the same. And so for each of these, as you're studying them and going through the different examples, jot down notes of what the pattern looks like so that when you get to the final exam, you'll look for the patterns, okay, for each of these. And not all of the exam questions obviously will be based on the different reactions, but you also need to know the information that you see in these slides as well. So don't just focus on the reactions, but also look at all the slides that have information. Those, I don't have to go through those, you could figure that out. But I just wanted to show you what these patterns are that you'll see on these different slides. Okay. And this one. Okay, so this is another, okay, I went through that one. All right, so this is another example. So in this example, you have your OH and your HOCH3. So you have two alcohols here. The hydrogen from one and the OH from your ring, excuse me, are going to form the water. Your OCH3 is going to connect directly to that carbon that's here at this point. And you're going to form your glycoside, okay? And remember, this is your hemiacetal alcohol that we looked at previously from week 6, okay? So it comes again, all right? And so we see this different example. And it's a hemiacetal alcohol because you have this portion over here, this alcohol that's already formed. And so it's like we're taking what we formed to the next level and add another alcohol to it. All right, and so then if we look at another example, this is with an amine. Remember, amines are always going to have that nitrogen present, okay, when it's connected to a carbon group. So amines always have, you're going to look for that nitrogen for amines. So the hydrogen that is connected to the nitrogen, okay, one of them is going to form water with the OH. And so then the nitrogen is now going to connect to the carbon like you see here. But there were two hydrogens, but only one of them is going to form with the OH to form water. Okay. So again, with all of these, you want to look at where the structures are going or what's happening with these different structures. Okay, and these are just to give you examples of different ribonucleotides so that you can get examples of those. There's some with the base and the different examples. So know these examples, okay? They go through the major bases, which are your purine bases, adenine, guanine, cytosine, thymine, and uracil. Those you see in the lab as well when we went through that with the letters, okay, for those particular organic bases, okay, that represent those. The letters are generally the abbreviations that are given, especially when you're looking at DNA and RNA sequencing. You see the letter more so than you see the full name. It's just our way of abbreviating it. So make sure you do know the name as well as those symbols for that information. All right, so that is the different weeks. So I did want to go through, and so as you're going through these, okay, which I strongly suggest that you start looking at it this week, refreshing your memory from starting with week 5. And if you are needing refreshing your memory for the functional groups, then I would suggest Googling organic chemistry functional groups. Like I said before, generally they're charts, okay, that you can find that'll have a structure and also the name of the functional group so that you can just refresh your mind, make sure that you understand the different functional groups so that if something said an aldehyde or if it gave a picture of the aldehyde, an example, you would know that those mean the same thing. Okay. And so I hope this was helpful. First of all, before I end this, I want to ask if you guys have any questions for me.
No questions. You've been very informative.
Oh, good. I'm glad. All right. Well, then, if there are no questions, then I wish you both the best and the rest of the class as you watch this recording a little bit later. If you have questions as you're preparing for the final exam or as you're finishing up week 7's information, please let me know. Make sure you check out the video for the lab for this week. I did post a video for that to make sure that you understand how to do the lab. It's a lot of information that's given in that lab, but for the actual portion where you're doing not just answering questions, but you're doing the experimental portion, watch the video because I went over how to do that. And then if you have questions on that, please let me know.
Okay, perfect. Thank
Um,
you.
thank you. Have an awesome week.
You too. You have a good week.
Thank you.
Thank you.
Bye-bye.
Bye bye.
Oh, you're welcome, Yvette. Thank you so much. If you have questions, let me know.